Hi, welcome to Lecture 6 in Social Neuroscience. What I'm going to do today is cover sort of a hodgepodge of leftover topics from covering the introductions of the nervous system and the endocrine system. What we're going to do is look at some relationships between the two in more detail, some sort of complications, things that make it more complex than it might seem to be at first blush. And we're also going to cover some other topics that are new in terms of how we learn and get affected by things that go on in our everyday lives. So these roughly map on to things that you cover in the Sapolsky book near the end of Chapter 2 and in Chapter 4. All right, so what we're going to do is, first of all, have just a quick review of the last 3 lectures, talking about the nervous system and the endocrine system, some of the key things I wanted to bring up. And then we're going to talk about how the nervous and the endocrine systems are more complex than you might think. We'll talk about Dale's principle, violations of Dale's principle, autoregulation, long-term potentiation, or LTP, and we're also going to talk about long-term depression, or LTD. And then finally, we're going to talk about the effects of sleep and sleep deprivation. All right, in terms of a quick review, I just want to remind you that, as we talked about a couple lectures ago, that we had our basic neuron synaptic communication here, where we have our presynaptic neuron, which has these axon terminals, and these terminal buttons. There's many terminal buttons for each neuron. And remember that each neuron could be having contact with up to 10,000 other neurons. So when we show here the dendrite of a receiving neuron, there isn't just one neuron that's receiving these signals. It could be up to 10,000 neurons that have their dendrites close by in this synaptic cleft. So where we have our terminal buttons releasing the neuron, you can see our little vesicles there are doing what's called exogenesis. They're spouting out their neurotransmitter into that synaptic gap. And then we have that postsynaptic neuron that may have receptor sites on it that bind to specific neurotransmitters. And this is sort of our classic depiction of what goes on in neural transmission, how we chemically stimulate the next neuron. And of course, if the dendrites of that next neuron get enough stimulation, they may cause an action potential to form at the axon hillock, and therefore cause another action potential to be going down its axon to another set of neurons. Well, anyway, this is our classic view, right? And so we have our action potential up at the top coming down and causing these vesicles to be released and their neurotransmitters. Now, going back to the endocrine system a little bit, I just did talk a lot about the hypothalamus and the pituitary gland, although there are many other glands that are going to release hormones. But one of the reasons why we focus a lot on the hypothalamus and the pituitary is they seem to be these master controllers coming from the central nervous system that really do control a lot what's going on in our hormones in our body. And specifically, I made this distinction here that in the pituitary gland, we have the anterior pituitary that's affected by hormones that are released by the hypothalamus. And then we have the posterior lobe of the pituitary that's affected by nerves and neurotransmitters that are also released by the hypothalamus. So there are 2 different ways here the hypothalamus could be affecting the pituitary gland, either through hormones or through nervous action, through neurotransmitters. And to just show you this in a little bit more detail, I found this. And so you can see here we have this very small little localized circulatory system that's known as the hypophyseal circulatory system. It's depicted here in these blue lines. And you can see that what's happening is up in the hypothalamus, hormones can be released by nerves that are up there. And then these particular hormones can then easily pass down through that circulatory system to the pituitary glands. And then in the pituitary gland, you can see there's all these different endocrine cells there that have capillary beds right next to them, so that the hormones can have their effects right on those particular cells of the anterior pituitary, and then cause them to release hormones themselves, and those hormones can then go out in the veins down at the bottom. So it's a very complicated little system there just between the hypothalamus and the pituitary gland for hormones to be released by the hypothalamus to the pituitary gland. And then likewise, the other thing to notice here is that we also have that posterior lobe, which is, you can see the green nerves that are coming down there. They have their nuclei up in the hypothalamus, and they're going to release neurotransmitters, and those neurotransmitters down in the posterior lobe of the pituitary gland are going to cause hormones to be released into the blood. And so you can also have hormones that are released from the posterior lobe of the pituitary gland that are also going to then get into these veins and then spread out throughout the rest of the body. So this very complicated system, and as I mentioned last time, we have different hormones going for the anterior lobe, and we have different hormones that are released by the posterior lobe of the pituitary gland. Now, I want to go ahead and just talk a little bit more about how the nervous and the endocrine systems are even more complex than you might think. Now, I know you're probably thinking this is already pretty complex, but they even get more complex when we start to realize that they don't just do the things as simply as we might think they would do. Now, one of the reasons why we think these things are more simple than they are has to do with something that's known as Dale's principle. Now, this is Henry Hallett Dale. He won the 1936 Nobel Prize for work on acetylcholine as a neurotransmitter. At the time that Dale was working on the nervous system, they only knew of 2 neurotransmitters. And so he was one of the first people to really show how these neurotransmitters worked. And as a result of his work, a principle arose, not necessarily stated by him, but by people who followed after him, that basically stated that a neuron performs the same chemical action at all of its synaptic connections to other cells, regardless of the identity of the target cell. So what this is saying is that each neuron basically performs the same chemical action no matter what, okay? Now, this principle, called Dale's principle, has sort of 2 versions that you'll hear about in the literature, and it depends on who you talk to, which books you look at, which lectures you listen to, but there are kind of 2 versions of this principle. One version is that a neuron releases the same neurotransmitter at all its axon terminals. This is kind of what was implied by the original stating of the principle back in the 1930s, that keep in mind that back then they didn't know about very many neurotransmitters yet, and it seemed that each neuron only released one kind of neurotransmitter. So if the action potential fired and it reached down there at those axon terminals, the terminal buttons would be releasing just all of them at the same time, the same neurotransmitter. We'll call that version 1 of Dale's principle. There's another version of this principle, and that is that a neuron releases its neurotransmitter or transmitters at every axon terminal at the same time. Now, it's going to turn out that we pretty much know that version 1 is false, and there's a lot of people who do subscribe to version 2, although there is some evidence to show that version 2 may be incorrect as well. So let's take them one at a time. Let's take the first principle, or the first version of the principle, and I'll just say right now that version 1 is certainly wrong. And this has to do with the fact that there was a discovery in the 1970s and 1980s of some neurons that actually release 2 or more different kinds of neurotransmitters. And this is known as co-transmission. So what you can see here are just 3 different examples of how this might work. This comes from this review paper in Current Opinion in Neurobiology. But you can see here that what it's trying to show you is, like in the simplest example, on A on the left, you could have a neuron that in its vesicles is actually holding 2 different kinds of neurotransmitters, glutamate and GABA, for example. And so when it fires its action potential and it comes down and causes those terminal buttons to release, it's actually releasing 2 different neurotransmitters that could then bind with the receptors on the dendrite of the postsynaptic neuron. And so you can imagine that now that we have 2 neurotransmitters possibly being released at the same time, that means that we could have more specific effects on the different neurons that follow it, that come after that presynaptic neuron, depending on where its receptors are, what receptors it has, and so on. You can also see that we can also have other kinds of combinations of neurotransmitters. The second example shows you what are called monoamines. Maybe here we're going to have a fast-acting neurotransmitter like glutamate that's going to go ahead and affect, again, binding of the dendrites there on the postsynaptic neuron, but it also could have a neuromodulatory effect. That is, it can go ahead and cause changes to occur in the way that the neurons fire. And then finally, the last one's trying to show you that you could also have neurotransmitters that bind in the postsynaptic neuron, where it actually goes and causes changes in the actual structure or the function of that postsynaptic neuron, and it therefore is causing changes at the nucleus level. So anyway, the main point here is just wanted to show you that Dale's principle version 1 is certainly wrong. We now know that there could be 2 and maybe even sometimes 3 different neurotransmitters being released by neurons. What about version 2? Now, remember, version 2 basically says that when the neuron fires, all the axon terminals are releasing the same neurotransmitter or neurotransmitters if co-transmission happens as a result of an action potential. So remember, we get our action potential formed at the axon hillock. It travels down along the axon, making use of the myelin sheath to speed along things. Then it gets to those branches of the axon, of the axon terminal, and all those branches all have terminal buttons there that could be connecting up to 10,000 other different neurons, right? So in the classic model of all this happening, that action potential, when it reaches all those different branches of the axon terminal, it's going to cause all of those buttons to release the same neurotransmitter or two neurotransmitters exactly at the same time at all places all at once. But even though that this is probably true most of the time, there are some exceptions. Already we know that it seems like that some blockage can happen where you can actually get some blockades that prevent the action potential reaching some of those branches. That is, the neuron itself can regulate where that action potential flows down into its branches, causing some of the branches never to receive the action potential, therefore not releasing their neurotransmitters. So that means that not all of the axon terminals would be all releasing all of their neurotransmitters at the same time. It could be that some of them are holding back and some of them are releasing. This is a fairly interesting wrinkle on version 2. And I actually tried to find some evidence of this myself. It's hard to find where in the literature this has been established. All I can really do is refer you to a lecture that Robert Sapolsky gives on YouTube when he was lecturing to his course at Stanford. And that particular lecture, he does mention this and says it was discovered back in the 70s and 80s, but I have yet to really see strong documentation about how this works. So this might be something that's fairly cutting edge and maybe requires more research. I just don't know much more than that, except to say that version 2 is probably correct most of the time, but there may be some chances here where some some moments where axons don't actually all release their neurotransmitters at the same time. Now, all of this stuff about Dale's principle applies to the nervous system, but we find that hormones have sort of similar complications. There's some parallels in the way hormones work in the endocrine system, and I just want to kind of show you how this works. For instance, the endocrine system can have co-transmission too. And so to give you an example of this is a much more detailed drawing of how the hypothalamus can affect what happens at the pituitary. And what I want to show you here is, remember, if we're thinking about just the stress response, the main thing about the stress response that we remember from looking at the HPA axis is that pituitary gland, if it gets the stimulation from the hypothalamus, is going to release ACTH out into the bloodstream, and then the ACTH is going to affect production of the adrenal medulla, which it's going to release cortisol and other stress-like hormones, right? So we get our stress reaction because of the ACTH that's being released by the pituitary gland. But it turns out that the hypothalamus has multiple ways of affecting the release of ACTH. It's not just simply, as I mentioned in the previous lecture, just CRH that does this, corticotropin releasing hormone. It turns out that stress is actually multifaceted, and therefore the hypothalamus can release different hormones that encode a stress signature to fine-tune our body's response to stress. Now, I said that CRH is the main hormone, but other hormones that could be released by the hypothalamus into that small little circulatory system could also include vasopressin, oxytocin, epinephrine. All of these things can be affecting what happens at the anterior lobe of the pituitary and cause the changes in the way that ACTH is released in the pituitary. Now, why might that be? Why would we get subtle little differences in the way a stress response is encoded through the hormones and how it affects the ACTH production? Well, if you actually look at how ACTH is then released into the bloodstream, there could be different secretory curves. That is, the way the secretion of the ACTH happens could be, for example, really short. It could just be a short burst of it that has a fast effect. So maybe a whole lot of it just dumped really quickly. Or it could be that it's a long, slow process that makes the ACTH linger at smaller levels that slowly build up. And whether it's a short, fast versus a long, slow distinction there would be determined by the pattern of these hormones that are released by the hypothalamus. So the point here is that it's not just one hormone from the hypothalamus that then affects the pituitary, which then affects the release of ACTH. I'm trying to argue here that you can actually have different patterns of these hormones being released at the pituitary that cause ACTH to be released differently depending on the kind of stress. Similarly, you can also have localized blocking of the hormone release. And the reason why this happens is that the pituitary, particularly the anterior pituitary, the anterior lobe of the pituitary gland, has really a mosaic pattern of cells that produce just one kind of hormone each. So you can kind of see that in this drawing here where it says endocrine cells. And you can imagine that we have tens of thousands of these endocrine cells. They're all neatly packed in at the anterior lobe of the pituitary gland. But each endocrine cell is releasing a different hormone into the bloodstream. And they're not just all, like one part of this is all of them clumped together that releases, ACTH, another one's doing something else. Instead, each one of those cells is releasing just one kind of hormone, and they're next to other neighboring endocrine cells that are releasing a completely different hormone. So even though blocking doesn't actually happen here, what can happen is that the pituitary can be stimulated, so just one neighborhood of these cells release their hormones, and then another neighborhood of cells somewhere else in the anterior lobe of the pituitary are not releasing their hormones. And then you can have this sort of local coordination between different cells and regulating the pituitary output. And remember, the pituitary is releasing all sorts of different kinds of hormones. And as it does this, it can release the different hormones, again, depending on which of these little neighborhoods of endocrine cells are currently being affected by the hypothalamus. So the point is, again, it's not just all of this happens at the same time, that all of the cells are going to fire. Instead, what happens is individual little areas of these cells can be firing based on what the blood flow is coming from the hypothalamus, and therefore you can get some much more specific patterns of hormone release by the pituitary gland. So it lets us have a little more fine control of exactly what hormones are being released at any given time. Much like what I was trying to say to you about that version 2 in the nervous system, that maybe some of those axon terminals are being blocked by the neurons so that they're not all firing at the same time. Same thing here is you can have hormones coming from the hypothalamus that go to the pituitary gland and only affect certain endocrine cells at a particular location that cause then only a certain pattern of hormones to be released at any given time. So it's not just this sort of like blunt edge thing that just happens all at once, that every time our hypothalamus fires, just one kind of hormones being released. The hypothalamus and the pituitary are basically working together here to have very fine control over which cluster or which pattern of hormones are being released at any given moment. Now, one last thing as we leave these sort of complicated things about the nervous system and the endocrine system, and this doesn't have anything to do with Dale's principle, and that one more complication here is autoregulation. So you might have noticed, and you've heard about it probably before, that neurons actually have autoreceptors on the axon terminal. And you can see this here. That weird looking thing that's sort of sticking out there in this diagram is an autoreceptor. And what that means is that the presynaptic neuron actually has receptors on it for the very neurotransmitter that is being released, which seems kind of strange. There it is, action potential is happening. Serotonin, for instance, in this example, is going to be released, and then there's serotonin receptors in the receiving cell. But at the same time, the presynaptic cell there at its axon terminal has little receptors that bind with serotonin themselves. So these autoreceptors, that's what they're called, provide measurements. that are needed for negative feedback regulation. That is, they're kind of keeping track. They're like a bean counter of how much of the serotonin is being released in the synaptic cleft. And then as the neuroreceptor gets that information back, it's sort of gauging how much serotonin is out there. And as more and more of that released neurotransmitter binds with the autoreceptors, the neuron can then get feedback that it should stop the release of the neurotransmitter. This is done. We've done our part here. We've reached the threshold of how much of that particular neurotransmitter should be let out. This happens all over the nervous system, and in different kinds of drugs that you can take can affect how autoreceptors work and how much whether or not they lower or raise the threshold of how much the neurotransmitter can be left out there in the cleft. Now, autoregulation also happens in the endocrine system. And so here you can see a sort of a classic example. It happens all over the endocrine system, but in this particular example, we can look at the relationship between insulin and glucagon. So here you can start where it says number one. So you eat a meal, it increases your blood glucose levels. So you've got food into your stomach, and now it's being digested, and now you're getting sugar flying into your bloodstream. And so eating this meal increases your levels to a higher than normal homeostatic set point. So your body likes to keep its glucose at a particular level. Now, because you've eaten, a whole bunch of glucose is coming in. That causes then the pancreas to release insulin, a hormone, into the bloodstream. So the pancreas is now cranking out insulin, this hormone. The hormone is going to be then passed throughout the body. And one of the places that it goes is to the liver, which it tells the liver to begin taking up all of that excess glucose and start to convert it into glycogen. Glycogen is like a reserve for our sugar, and it's going to go into our fat cells. We're going to store that glucose, that excess glucose now for future use. So it's like putting it into the bank. Okay, and as the liver does this, it changes that glucose into glycogen. It's going to cause the glucose itself in the blood to drop to normal. And as it starts to go down, what we can see then is we're going to get back to close to the set point. But now imagine that you have gone to a point where you've not eaten for a while, so you're getting kind of hungry. And you can see there in the green where it says number one, missing a meal brings the glucose levels down below the normal set point. So now you haven't had enough food, and your glucose level is now below your set point. What happens is the pancreas is going to, again, release a hormone into the bloodstream called glucagon. And the glucagon goes out into the bloodstream, and one of the places, again, that it targets is the liver. And the liver begins to then convert all that stored glycogen into glucose. And then the glucose can be used to go out into the body to give energy to all of the cells that need it. And so we can get back up to the normal set point. So this happens constantly through the day, that you got this relationship between the pancreas, the liver, and these hormones of insulin and glucagon happening as a result of autoregulation. Now, autoregulation happens naturally, but you also will find that a lot of different disease states that happen in the body have to do with problems of autoregulation. Basically, where we go too high or we go too low, and from our set points, and our systems can't handle that very well, and then you can start to have a breakdown that causes disease states. So just to give you an example, go back again to the nervous system and talk about autoregulation. Well, depression is an interesting multifaceted problem when people suffer from clinical depression. And there could be several abnormalities that are involved with this, and in terms of neurotransmitters, and those would include things like serotonin, dopamine, norepinephrine. But what happens is if you are then treated for depression with a standard antidepressant like Prozac or fluoxetine, you get changes then in the blood concentrations of the neurotransmitters in just a matter of minutes or hours. So what happens is part of the effect of these antidepressants is they just change the levels of those neurotransmitters to get them closer to whatever the set point should be, because you possibly have some abnormality about where your serotonin, dopamine, and norepinephrine are. And you can get those changes, like I said, very quickly. So you take the drug, within a matter of minutes or hours, you start to see changes in the levels of the neurotransmitters in the person's brain. However, a lot of people report that when they take their antidepressant, that they get no relief from their depression, that it takes a long time, actually. Even though we can document that the neurotransmitters have changed, we're not seeing relief then from the problems of depression. So they don't start feeling better, often for days or weeks. Why might this be? Well, it perhaps has to do with autoregulation, perhaps because the autoreceptors themselves need to change and then change the autoregulation, which takes a longer time. So perhaps the reason why the person has been suffering from depression for a long time is because the autoreceptors are off in terms of what they think should be the proper amount of the neurotransmitter being released out in the synaptic cleft. And because of that, because the autoreceptors themselves are mismeasuring or miscounting the amount of the neurotransmitters in the cleft, what's going to happen is as you change the amount of neurotransmitters out in the synaptic cleft, the neuron itself is going to have to change its autoreceptors. It's going to have to structurally change them to better account for the amount of neurotransmitters there. So it's going to take a while to structurally change the autoreceptors in the presynaptic neuron so that it properly handles the right amount of neurotransmitters out into the cleft. So that's probably why it can take weeks before you actually see a real change in the way a person feels, even though the neurotransmitters have actually changed. All right, that's an example in the nervous system. What about in the endocrine system? Well, I can give you the example of adult onset diabetes. Normally, as we just talked about, the glucose in the bloodstream is being detected by the pancreas, and it in turn releases insulin, the hormone, which then signals that the fat, the liver, and the skeletal muscle cells should take the glucose up, right? So we just got over that. That's the normal thing that should happen. But what if there is no capacity for instance, these fat cells to take up more glucose? Which is actually common in overweight people. They basically have maxed out their fat cells in their body. And so what's happening is when all that glucose is in there, you're going to get this signal from the pancreas, hey, I'm sending out insulin, please liver, start to convert all of this glucose and store it. But basically the cells that normally would respond to having insulin in it, that would take up the glucose, are basically shut down. They're like, got too much already, we can't do anything. And so what happens then is the glucose just stays high in the bloodstream. And as it releases high in the bloodstream, that means then the pancreas just keeps cranking out more insulin because it's detecting there's a lot of glucose there. It doesn't know why. So it just keeps releasing more and more insulin. And what's happening is your body, the rest of these cells that normally would take up the glucose, have developed resistance. The person's developed insulin resistance, that the insulin no longer signals to them that they should actually take up more glucose. So autoreceptors in the pancreas then cause it to increase more insulin, as I said, and eventually the pancreas works so hard because the autoreceptors keep telling it to create more insulin that you end up burning out the cells in the pancreas that produce insulin, and therefore you've got poor insulin production from that point on, which causes then adult onset diabetes. So basically you've just stretched your pancreas so much by that increased insulin resistance that the pancreas can't produce insulin correctly then in future meals when you have glucose. So like I said, you get these disease states probably because a lot of what's happening has to do with autoreceptors and autoregulation. Okay, that's in terms of the topics, in terms of more complex issues about the hormone and the nervous system. I want to now move to a different thing about the nervous system itself and just talk a little bit about long-term potentiation. So this is, again, one of these sort of grab bag topics here that you'll get in one of the chapters in Sapolsky that have to do with how we actually change in our nervous system as a result of experience and learning. All right, so we're going to talk about this topic of long-term potentiation. And this really has to do with memory and how we store these experiences. And there I've got a picture of Donald Hebb, who did some very important pioneering work on the storage of memory in our nervous system. So this has to do with this question. Why do some things that we experience stay in our brain the rest of our lives, and other things are just more fleeting? They're gone just minutes later. How can we explain that with the way that the neurons are functioning? How do we learn is really the basic question here. Now, some people might answer, if you just ask them out in the street, how is it that you form new memories? And they might say, well, it's because we make new neurons to encode these new memories. All right, so the idea is that perhaps every time you've got some new skill that you learn, or you've now had a new event in your life, that you've had some new neurons being formed that encode those memories in the neurons. But the answer to that is no, that doesn't actually happen. Adults don't tend to make new neurons. There is some exceptions to this. We're going to talk about it in a future lecture. But for the most part, no, we don't actually make new neurons to encode new memories. Well, perhaps what it is then is if we don't make any new neurons, what we do is we make new synapses with the existing neurons. So we already talked about how each neuron has up to 10,000 connections with another neuron, with other neurons. Perhaps it just adds another one, 10,001 synapses to another neuron. Nope, the problem with that is, according to Hebb, that doesn't happen. So Hebb, his big important contribution was he argued that no, we don't actually make new synapses with existing neurons when we learn. Instead, what we do is we strengthen the pre-existing synapses. Okay, so we're going to make the connections that are already there between these neurons stronger as a result of learning and experience. All right, so how does this happen? How do we get learning happening then in the synapse? So when a presynaptic neuron fires many times in a short period of time, it causes a stronger response in the postsynaptic neuron. And the key neurotransmitter for this, to have this take place, is called glutamate. It's an excitatory neurotransmitter. So how glutamate is involved in learning is really detailed nicely in Sapolsky's book in chapter 5. And so I'm not going to go into it in great detail, but essentially the dendritic branches that are responsive to glutamate have two kinds of receptors. So in this drawing here, you can see this is actually in the receptor of the dendrite, the dendrites in the receiving neuron, okay? And so here we've got in this dendrite two kinds of receptors, one called MDA, and the other is called non-MDA by Sapolsky, but you can also call it here AMPA. It doesn't really matter, but we have these two different kinds of receptors, NMDA and AMPA. And if only just a little bit of glutamate is being released into the synaptic cleft, it's going to bind with the AMPA receptor, that green one. All right, so just a little bit gets bound to the AMPA receptor. And if there's just a little bit of these AMPA receptors getting it, it's just going to contribute a little bit of excitation in the dendrites, and you're not necessarily going to get an action potential. However, imagine now, though, that these AMPA receptors are just overexcited, and there's just a lot of glutamates being released by the synaptic cleft, okay? So if you've got a lot of this, a lot of excitation, all the AMPA receptors are all firing now, what happens is when this happens over and over again, the MDA receptor, which has like a gate, opens up and it lets glutamate bind to it. So the AMPA receptor is being excited by all of this extra glutamate in the synaptic cleft, and it's causing then the NMDA receptor to become open, and it allows then more glutamate to come into it. And then when the glutamate binds to it, there's basically an explosion of excitation, and the neuron's going to fire an action potential. All right, so this is the key to how this sort of long-term memory is going to take place, is that if there's just a little bit of excitation coming from the prior neuron, the presynaptic neuron, the AMPA is just going to keep firing a little bit, and it's going to cause a little bit of change. But if the AMPA starts to detect a lot of a lot of glutamate, then it's going to cause the MDA receptor to open up, and a lot of glutamate will then bind to NMDA receptors and cause the dendrites to create enough of a potentiation, enough of a change at the axon hillock to have an action potential go off. So here's what that's called, by the way, is called long-term potentiation. So this is going to cause changes in the receiving dendrites. So if this keeps happening, all right, so if you keep getting a lot of this happening over and over again, where the AMPA receptors get overwhelmed and they cause then the NMDA receptors to open up and let this influx to happen, what that means is that eventually we're going to get an increase of sensitivity. So you can see here what this is trying to show, that in a normal synapse, perhaps we have our our normal NMDA, they're not really getting much happening to it because there's not a lot of excitement coming from glutamate. But when there's a lot of glutamate happening, then eventually the AMPA receptors are going to have more of them, are going to be formed by that neuron, so that it becomes even more sensitive to having stimulation from glutamate. And if the AMPA receptors change and there's more of them, that means then it's going to be more likely that this postsynaptic neuron is going to fire when glutamate is released. So that is how we strengthen a connection, or strengthen the connection between two neurons when we're learning, is through this process of AMPA and NMDA. This changes over time because of repeated firing of this particular combination of these two neurons. Okay, and that's known as long-term potentiation. There is also a complement to long-term potentiation. It's called long-term depression. This is not related to clinical depression. It just means between these neurons, you can have sort of this opposite effect happen, which basically means that if there isn't much firing going on between that presynaptic neuron and the postsynaptic neuron, that there isn't much glutamate being released at all, we're not going to see any of those AMPA receptors happening. They're not going to be fired very often, so there's going to be fewer and fewer of them, and basically AMPA receptors become downregulated. That means this postsynaptic neuron is hardly ever going to fire. And it's not so much that you're, that it's like the whole connection is just becoming weak. It's more like you can consider this to be sort of a more efficient way of learning. It's like letting us know that this particular pair of neurons isn't involved in this memory or in this particular experience. And so they're de-emphasized in this pattern of neurons that are going on for whatever the kind of learning that's happening. So long-term depression is a way to basically get rid of connections that are in the way. All right, they're kind of like the ones that are superfluous and are not going to really help very much in learning what's happening. So it's really this combination of LTP and LTD together that's going to allow us to learn what a response is, or to have a memory of an event, or what the capital of the ACT is, or whatever it happens to be. Right? So just a little bit more about LTP, that LTP effects happen prominently in the hippocampus. You remember the hippocampus is where we have a lot of our memories being stored, but it really does happen actually all over the brain. So for example, it's going to be involved in parts of the brain that have to do with motor memory, learning how to move your hand to pick up a bottle or whatever it is. You get that encoded so you don't have to each time figure out how do I pick up this bottle. That is going to happen in other parts of the brain outside the hippocampus. Another example would be fear learning in the amygdala. There was a lot of discussion about this earlier when we were talking about the amygdala, how it helps us learn what things we should fear, what things should be threatened, that are threatening. And so the LTP effects can happen there as well. So it's not just in the hippocampus. LTP can be adaptive, but it's also can be maladaptive. So for example, in the cases of PTSD, which definitely implicates the amygdala, we can have sharp learning that occurs that it's really hard to unlearn. So a fear response to certain loud noises could cause a person who has PTSD to keep having really big stressful responses to loud noises, even though there's no danger anymore. And it's because basically this strengthening happened at that particular place where the PTSD was caused, and therefore it's now made this really strong connection between those neurons, and LTD hasn't actually happened. There are 3 ways that you can disrupt LTP. So if you are interested in how we can prevent LTP from happening, this is interesting. So one of the ways that this happens is through hypoglycemic states, that basically when you're hungry, your insulin's dropping. You remember we talked about all this happening here where you can get hungry and your whole body, your pancreas and your liver are changing. Well, it turns out that as insulin drops, this is actually where LTP drops. So like in the hippocampus, there are receptors there that can actually tell that the hormone insulin is lowered. And therefore, the hippocampus isn't going to be able to learn as easily, or because it's not going to be able to do LTP as much. So that's not a good time to learn something when you're starving. It's kind of like saying that during this particular time when we're really hungry, we're not going to be so much interested in learning new things. We just really want to get ourselves fed. Another way that you can disrupt LTP is through stress. Glucocorticoids that are going to be released there by the adrenal gland do improve LTP for the tiniest details. Okay, so it turns out that when you're under stress, the glucocorticoids do feed back to the hippocampus, and the hippocampus can detect that you're under stress, and it's going to actually facilitate learning for the tiniest details. So you can imagine, like if you were in a car accident and you have this detailed memory of what happened in the car accident, like it almost goes like a slow motion movie as you think back to the car accident. That has to do with the fact that those glucocorticoids that were released during that stressful event improved your LTP and made it happen very quickly, that you made those connections in your neurons about that particular event. But the problem is if stress is chronic, Like if you're chronically having high glucocorticoids, it can damage LTP and make it difficult for you to learn new things and, in fact, affect memory. So stress has a short-term effect of improving LTP for tiny little things, but if the stress is chronic, you're just constantly in a very stressful environment, that can damage LTP, prevent it from happening, and therefore affect memory. And finally, I can just mention alcohol. Alcohol also disrupts LTP, and this is because alcohol does cross the blood-brain barrier. So when you drink alcohol and it's in your bloodstream, it's going to basically get washed through your whole brain there, and having that alcohol disrupts LTP. So it's harder to learn things while there's alcohol in your system. Okay, so those are things that we need to know about LTP and consequently LTD. Just overall then, go back to that question about how is it that you learn different things? How do you know what you know? Keep in mind that each neuron is connecting to at least 10,000 other neurons, right? So that would mean then small changes in the LTP or in LTD start having large effects. So if we're having just a few of those 10,000 neurons being affected by LTP, that could be enough to cause a change, a large effect in the network of neurons, because our neurons really are connected in these vast networks. And so if you get little bits of changes here through LTP, that could be what causes the changes then that lead us to having memories and more complicated behaviors. So we start to learn when networks of neurons fire at the same time. So memory is really encoded in a neural network, not just a single neuron. It's not going to be just between one neuron and the next neuron. It's really the fact that this is happening over tens of thousands of neurons all at once that we get a neural network. Okay? And then why do we have some memories that are stronger than others? Well, our memory variation comes from, first of all, the amount of glutamate that was released at the time. So keep in mind just that basic process of how much glutamate is being released in the synaptic cleft is going to determine whether or not we actually get LTD or LTP. And then how strongly the postsynaptic receptors responded to it. And finally, how the neural networks are actually constructed. So all of that comes into play about whether or not this particular instance is all it takes. Like this one time you had the car accident, that's all it takes place from the memory. Or is it going to be that you have to learn this Chinese character when you're taking a Chinese language class, that you're going to have to kind of do this with flashcards over and over again before the amount of glutamate is going to be strong enough to cause a real LTP, and you're really going to start to learn those vocabulary words. So it all varies depending on these three factors here. All right, that's LTP and LTD. The final topic for this lecture is I want to talk about sleep effects. And this isn't really talked about by Sapolsky in his book. But I think that sleep effects you're going to see really have profound impacts on our social behavior. And so I just wanted to talk a little bit about sleep as it might pertain to social behavior. Now, if you've covered sleep in previous courses, you might remember that there are kind of two broad kinds of theories about why it is that we sleep. And one broad theory about why we sleep are known as recuperation theories. Basically, the idea here is that being awake disrupts our homeostasis. So our natural state is that we'd like to be really still and calm. And when we're awake, we're disrupting our homeostasis. And so we're using up more energy, more things are going on when we're awake. And therefore, we have to have sleep to countervail that, to counteract it. So sleep restores our energy levels and releases the toxins from our brain that happened while we were awake. So those would be the recuperation theories. There are these other theories, though, out there for sleep that are called adaptation theories. And they focus more on that they say we sleep because of a 24-hour timing mechanism. So we actually kind of keep track of the 24 hours in a day. And why we sleep, why it evolved that animals sleep, is perhaps just to conserve energy when we don't really need to be doing anything. So basically, our bodies want to conserve energy most of the time. But of course, there are times when we need to have energy, like to go and get food, to hunt, to have sex, or whatever it is. But when we're not doing those things, then the best thing might be to do just to conserve energy by going to sleep. And so the idea here is that sleep isn't really necessary to stay healthy. It's not doing anything to restore our energy levels or our toxins. It's really just like basically timeout, saying, hey, we don't have to be doing anything else right now. and our body tells us it's that time of day where nothing's really going to happen. We're not in danger. We're not going to have sex. So it's to go to sleep. So that's the adaptation theories for sleep. Now, some brain areas that are involved in sleep include the following. And so I have here a nice diagram that shows you all of these different parts that are involved in sleep. But I only want to focus on some key areas here. So our old friend, the hypothalamus, is definitely involved, right? So the hypothalamus is involved in both wakefulness and sleepiness. It's involved in the sense that we have the posterior and the anterior parts of the hypothalamus involved in wakefulness, and then the anterior parts involved in sleep. But it's also the location of what's called the suprachiasmatic nucleus, or the SCN, which is the brain's master clock, and it contains neurons that fire in a 24-hour cycle that much matches the adaptation theories of sleep. The SCN is really controlled by signals from the retina, which give it light during the day, and by melatonin or melatonin, depending on how you want to pronounce it, in the pineal gland at night. So it keeps track of where you are in the day cycle based on the light that's coming from the retina and by melatonin that's being produced by the pineal gland at night. And then we move down more into our reptilian brain and look at the pons and the thalamus. And we can see here that the thalamus is number 2 in the diagram and the pons is number 1. Well, the pons produces acetylcholine during wakefulness, and then this acetylcholine activates the thalamus, and the thalamus then sends signals off to the cerebral cortex that makes us conscious and awake during the day while or night, or whatever it is, when we're awake. So that is another important part of this. And then finally, we have the VLPO, the ventrolateral preoptic nucleus. And basically, as the brain runs lower in energy during the day and gets near the end of the day, it triggers activity here. Neuron activity happens in the VLPO, which then sends neurons back to the hypothalamus and the pons. And they subsequently, these neurons that are being sent to the hypothalamus and the pons, are being influenced by what's going on with the SCN. So if you keep track of the SCN, it's kind of like telling us what part of the day it is, whether it's daytime or nighttime. It's also feeding into these signals that are coming from the VLPO. And as that all works together, it's basically going to cause a shutdown in our brain and we go to sleep. So those are the main areas that are involved in sleep. Now, I don't want you to focus too much on this. We're not really in a course here where we're trying to understand how sleep works. I just wanted to show you how important the hypothalamus, for example, is in all of this. And if we already talked about how the hypothalamus is involved in the pituitary gland and all of these hormones, as well as what's happening in terms of regulation for the autonomic nervous system, you can see that sleep then is going to have a very central role in affecting our behaviors. All right, so let's look at sleep deprivation and how it affects our social behavior. So what is exactly sleep deprivation? Well, the way that you want to think about sleep deprivation is this definition I have here, that it's just achieving less sleep than is required to function. Now, I say it that way, not in terms of how many hours of sleep you should get. That's because at different ages, people have different amounts of sleep that are required. When you're younger, you need a lot more sleep. When you become middle-aged, you have less sleep. And then when you get older, you might need more sleep again. But the point is that whatever age you're at and whatever your activity levels are, you have sort of like a set point of amount of sleep that you need. And for most people, that's on average around 7 to 8 hours of sleep. All right. So if you have a sleep disruption, something happens in your life that causes you not to get that normal amount of sleep that you're accustomed to having, we call that sleep deprivation. How do you study sleep deprivation as a researcher? Well, there's 2 ways you can do this. One is you can look at it in the laboratory where you go ahead and ask people to give up sleep. So, for example, if somebody gets normally 8 hours of sleep every day, what you could do is you could tell them that for the next 2 nights, I want you to stay up till 4 in the morning and then get up at 8 in the morning. So you only get 4 hours of sleep and then come to my lab. So you disrupt the sleep just through instruction. And you can use diaries and cameras and things like that to make sure the person's really disrupted their sleep. The other way you could look at sleep deprivation is just through people who are naturally sleep deprived, that have disrupted sleep because of what they do for their job. And this would include, for example, shift workers, people who work in healthcare, who work in assembly lines, all sorts of jobs where they're not working during like sort of the normal 8 to 5 hours. They work late night shifts. This week they work these shifts. Next week they work different shifts. So this would mean that their sleep is constantly being disrupted. They're not having a normal pattern of sleep very easily. So what we know about that sleep deprivation is it does affect self-regulation. So when people have their sleep deprived, they don't get their normal amount of sleep, a real simple thing that it does is it affects self-regulation. And you can see this self-regulation, I'm going to tell you about this in terms of social behavior and emotion in a little bit, but we can take it really simply and just look at, for instance, eating behavior. That when you're sleep deprived, research shows that you're more likely to choose energy-dense foods, things that are sugary, fatty foods. It's very common when people are sleep deprived that they pick these less healthy alternatives for food. Why is that? Well, perhaps it's because of hormone signaling that regulates your hunger and your satiety. That is that when you know you're full, that whether or not you need to eat right now, all of that's impaired because of your sleep deprivation. Your hormones are not, maybe through autoregulation, whatever it is, it's being disrupted by your sleep deprivation. Some examples of what we know about this and how we know that people really do this, and has to do with work with shift workers. So for example, you can imagine air crew, like people who do flights that are international, they're crossing many different time zones. A lot of air crew, what they do is they engage in what's called preventative eating. And that's because they know they're not going to be eating at their normal dinner time or their normal lunch time. And because their schedules are so irregular, they just get their food whenever and whatever they can eat. And so they end up eating all sorts of strange amounts of food, not necessarily healthy food, at really odd times because of the fact that they have this sort of strange time schedule. Healthcare shift workers, for instance, nurses, tend to use a lot of sugary foods, juices, and caffeinated drinks to stay awake. So you'll find that if they have to work the 11 to 7 shift tonight, they took it for a friend or to pick up some extra money, they're going to have methods that they use to kind of keep themselves awake. And so they tend to pick things like sugary foods and juices and caffeine to kind of help them stay awake during the night. So right there, we can already see some simple things that seem like healthy strategies to deal with the fact that they're shift workers is causing them to not necessarily eat very well and therefore disrupting maybe the endocrine system. And the other thing that's interesting is that there's research that shows that people who have disrupted sleep, people who are, for instance, shift workers, often reward themselves with food. So here you can see some qualitative research that was done by Dorian et al. In 2019, where they looked at shift workers and asked them about different kinds of food that they reward themselves with. And a lot of this is, by the way, quite social in the fact that the other people who are working that same shift take the same time off and they kind of celebrate together. And so there's a lot of social pressure on eating these more indulgent foods as a reward for doing their shift work. So you can see here someone says, I'll go down to the vending machine and get a chocolate bar like it's been a hard night. My compensation for being awake all night is to eat lots of junk. The CCC of night shift, chips, Coke, and chocolate. Night shift, well, that's a totally different matter. You have buns, biscuits, steal them out of the fridge because it's there, it's easy, and it's readily available. And then the last one, someone will bring in cake on Saturdays. Everyone feels sorry for themselves working on a Saturday, so you're pulled in to have cake. So the point here again is that people are not necessarily making the best healthy decisions about their food, and this could be related to the fact that they have disrupted sleep. Their self-regulation is a bit off. And what Dorian and colleagues have worked on here is to try to understand this in a more biological, psychological way. And what they do is they talk here about if you have a stressor and then you appraise that stressor negatively. So that is, you have you have to work this late night shift and you have a bad attitude about it. That bad attitude that you appraise is going to lead you to have a negative mood. It's going to cause you then to have more negative affect. It's going to cause you to be moodier. And because of that negative affect and that emotional lability of your mood, it causes changes in the HPA axis, which, as we know, will increase your cortisol. So you're going to have this stress response. But another thing it does in the HPA axis as a result of that mood is it's going to increase your ghrelin, which is a hormone that's produced by the GI tract that signals to your body that it's time to eat. So that's increasing, that hormone's going up as a result of this negative mood. And therefore, where does sleep loss come in? Well, the sleep loss is then going to dysregulate this. It's going to make it hard then for your frontal cortex, for example, to keep this in control, what's going on with the HPA axis. And so you might end up getting in this loop going around and around and around where you keep getting more and more stress, this negative mood's happening, you're going to eat more poorly. Also, because you're sensitive to the fact that you're eating these indulgent foods, your brain's reward system is going to cause endogenous opioids to be released, makes it like better to be eating these bad foods because you're in such a bad mood, and so you end up eating more indulgent foods. So our sleep loss is really having its impact largely because it's not allowing us to regulate our HPA axis, it's not allowing us to regulate our mood very well, and we're more vulnerable then to just simple brain reward activity. This increase of these rewards by eating these sweet foods. By the way, this not only works, this whole breakdown of self-regulation for food, it also works, for instance, with alcohol consumption. So this is another study that was done by Dorian et al. And what you can see here is that they talk about basically how much drinks people have. And so this was actually a study of Australian shift workers, and they found that the shift workers themselves drank the same amount of alcohol daily or near daily as anybody else who didn't have a shift. Okay, so it's not that people who choose to work in shifts drink more. It's nothing like that. They seem to drink the same amount of alcohol over like an average week. But they do tend to binge drink more when they are sleep deprived, when their sleep has been disruptive. And so you can see that binge drinking is, for instance, defined as having like 7 drinks at a time or 5 drinks for women on a single occasion. And so what we can see here is that 12-hour rotating shifts are people who are going to work for 12 hours straight and then get 12 hours off. And obviously, if you've been working for 12 hours straight, then you have 12 hours off, and then you have to come back on again, that's going to disrupt your sleep. And we find that those people in that particular study, which were in this particular case was 96% male, they were much more likely to drink many drinks all at once when they were in that sleep deprived mode. And then you can see other kinds of shift workers, for instance, people who work 8-hour shifts that keep changing all the time. So maybe this week they've got a night shift, next week they've got a day shift. That again, that causes more drinking above the normal number of drinks you should have at one particular point for both men and women. So the point here is that having sleep deprivation as part of your job may mean that you're going to end up doing more binge drinking. And there's also evidence, by the way, that shift workers use alcohol as a sleep aid. So when it's kind of stressful for them, they're having a hard time getting to sleep because they've just changed their shifts again. A lot of them actually drink more at that time, which then causes them to have binge drinking, and not necessarily a good thing for them and their long-term health. So this is a real problem for shift workers, probably because of the sleep deprivation that's happening as a result of their work. All right, let's move on to then finally talking about how all of this plays a role then in social behavior. Now, if you think about it, social behavior really does require a lot of self-regulation. And we've already talked about the fact that people are not really good at self-regulation about alcohol and food when they're sleep deprived. So if we look at how they regulate their behavior and their emotions in terms of social behavior, what happens? So shift workers, for instance, report a great deal of negative emotions. And this is often something that they talk about, and then they blame it on the fact that they have these shifts, these sleep-deprived nights, and where they're not getting enough sleep on their job. And then we have people in the laboratory who have their sleep disrupted as part of an experimental manipulation. And it turns out that they have more problems with executive control. So if you've gone ahead and disrupted someone's sleep, bring them into the lab. Executive control is largely in the prefrontal cortex, so we probably have disrupted their prefrontal cortex activations here. They do more poorly on tests, on neuropsychology tests that look at executive control, things like the Stroop test and the go, no-go task and other kinds of tests that you might have heard about before that have to do with executive control. People who are sleep deprived also have more lapses in attention that can make it difficult for them to be a good group member. Okay, so what this means is that people who are more sleep deprived can be making more mistakes. So for instance, example of this would be that nurses report making more errors when they had less sleep in the 24 hours prior to their shift. So they actually did this in a diary study where they had the nurses talk about each time they worked and what they happened at that particular job. And they asked the nurses to talk about any mistakes they might have made and so on. And then when they go back and analyze this, they find out that the nurses are reporting more errors when they've had disrupted sleep in the previous 24 hours. But one interesting thing that also came out of this was that they were more likely to catch other people's errors if they were sleep deprived. So that is, they're making more errors than themselves, but they're also more likely to catch other people's errors and blame them for the problems that they're having. And you can see how that could be a problem, because if you are making a lot of mistakes because you're sleep deprived, and then you go around and you see that other people are making more mistakes, or noticing that they're making mistakes, even if they're not sleep deprived, you're not necessarily going to be a good group member, because as a nurse working in this particular unit, you're making lots of mistakes because you're sleep deprived, and you're going around and pointing the finger at other people and blaming them for their mistakes. So this is what we mean by the fact that they have these lapses in attention that can make it difficult for them to be a good group member. So sleep-deprived people do tend to blame others more, and they tend to be more hostile. This was a study that was done in the laboratory where they had 26 healthy young people who were well rested, and that's what the black bar represents, is the baseline when they were well rested. And then again, after they've been awake for 55 hours. So they had to stay awake for 55 hours and come in and fill out a diary. And you can see that some big differences happened here. So up at the top, they were more likely when they were sleep deprived to direct blame and or hostility towards someone or something. They were more likely to minimize the frustrating situation to the point of denying its existence if they were sleep deprived. But down at the bottom, you can see that they're also less likely to accept the blame for a frustrating circumstance. They're regarding the frustrating situation as unavoidable and not blaming anyone. That was less likely to happen when they're sleep deprived. And they were insistently pointing out the presence of a frustrating obstacle when they were sleep deprived. And they were less likely to offer amends to solve the problem, usually out of a sense of guilt. So the point here is that these are just sort of normal people who've had their sleep disrupted, and you can see it's affecting how they feel and how they see different kinds of situations, how they appraise situations. And so someone who's sleep deprived is going to probably have more problems than getting along with other people, because they're blaming others for what's going on. They're, it's not a good state to be in if you're going to be in a social situation where you're more blaming and more hostile as a result of being sleep deprived. Sleep loss can also affect how the medial prefrontal cortex and the amygdala network works. And so this is actually covered in a nice article by Simon et al. In 2020. And what you can see here is they're talking about the amygdala. And remember that the amygdala might be involved or is involved in threat detection. And so what it's showing you here is in A, that if you just look at the amygdala for sleep-deprived participants versus sleep-rested, that people who are sleep-deprived show a lot more amygdala activity to different kinds of stimuli that could be threatening, or they are more likely to be more prone to be activated to different stimuli and to think of them as possibly threatening. So it makes your amygdala sort of like over responsive to any of the stimuli that are out there. And they argue that partly what's going on is that the medial prefrontal cortex would normally kind of come down and tell your amygdala, hey, don't worry about this thing, this is a safe situation. That's impaired with when you have less sleep. And so as a result of that, your amygdala is kind of like really dominating your responses. And so they show this here is that if you have a bunch of different faces that vary in how threatening they look, you can see that the one in the middle is the most threatening in that picture. Of course, if you were sleep rested, that'd be the only time that your amygdala would really fire. But the point here is that without the medial prefrontal cortex really having this chance to regulate what your amygdala is doing, you're more likely to see things that are more neutral also as threatening, at least that your amygdala starts to be more responsive to it. That's what it means by in the red where it's sleep deprived. So again, the whole point here is that if you're sleep deprived, you're probably going to be more responsive to stimuli out there that are threatening, but you're also going to view things that are not threatening as being more threatening. And this might have to do with all that blame and being upset with other people because you don't have the medial prefrontal cortex coming down and regulating your amygdala response. And how this might play out at work. They talk about this in the article that perhaps if you have a leader in your workplace who's not getting enough sleep, they're going to have increased hostility, they have increased abuse of their workers, then they have less charisma, less charm, because the prefrontal cortex isn't actually causing them to be very good at their social behaviors. If you're just an employee, you could show that you're not getting enough sleep, you're having increased hostility, you're going to have less or poor relationship quality with you and your boss, but you're also going to have increased hostility and less helping of your coworkers. And so it's a real problem if you have sleep deprivation here in terms of social behaviors and looking at productivity on the job. The final thing I wanted to say about all this is that Gordon et al. Has this really nice paper called The Social Side of Sleep, elucidating the links between sleep and social processes, that they published in 2017. And they try to put it all together in one big thing. And this is just to, again, just point the path to you to show you how complex and how rich sleep could actually be in terms of social processes. You'll notice that most of what I've been talking about is just the total amount of sleep that you get. But what they point out, the Gordon et al. Article points out, is that there are a lot of other kinds of sleep parameters that you could look at in terms of not only just sleep duration, but how quickly you fall asleep, when you wake up, all sorts of other, whether or not you're in REM versus non-REM sleep. All of those are variables that we don't really fully understand yet in terms of how they might affect cognitive functioning, how it affects your physiology, the autonomic nervous system, your endocrine system. And then those mechanisms then affect your social processes, and it could affect all sorts of things like your attitudes and persuasion, your close relationships, how you get along with people in other groups, your morality, and so on. And so this is all coming in together to show you that sleep could be very central to what happens on a day-to-day basis in terms of how people work with each other, how they get along with their close partners, and so on. So if sleep deprivation is happening quite frequently, this is going to be more disrupted and because it becomes a real big problem. So again, I'm just introducing the idea of how sleep deprivation could have a really vital role into how the nervous system and the endocrine system function. We'll come back to it again later. For instance, one of the first things we're going to be talking about is adolescence. And as you might know, adolescents have lots of problems with sleep. And so when sleep is being disruptive in an adolescent, who knows, with all these other things that are happening in the adolescent brain, how it's affecting their social behaviors. So in sum, we've talked about just reviewing a little bit about the nervous and endocrine systems at the very beginning, but then I talked about how the nervous and endocrine systems are probably more complex than you might think. We talked about Dale's principle, violations of Dale's principles, version 1 and version 2. We went through autoregulation and some of the problems of autoregulation. I talked about long-term potentiation and long-term depression in terms of how we learn and have memories. And finally, we reviewed the effects of sleep deprivation, how it affects self-regulation, and how it affects our emotions and social behaviors more generally. So that's it with my basic coverage of the nervous and the endocrine systems. From now on, the rest of the lectures are going to kind of move to other parts of social behavior. So what we'll be doing next is talking about development in the adolescent brain, and then we'll get into talking about genetics. Thank you very much.